Hello everyone and welcome to today's session, and today we'll be discussing the NCERT section 8.5.10 and 8.5.11. Okay, so this is the part 7 of the lecture. Before starting the lecture, I just want to give you one information that the images, text and other materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and you have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please know that parts of the content may have been generated using generative AI, which is experimental. So, what are the objectives of today's session? So, today we will talk about and we will understand the structure and function of the nucleus. See, nucleus is a very important organelle. Okay, it consists of the genetic material, which is DNA. Okay, so we'll be talking about the structure of the nucleus. We'll be talking about the function of the nucleus. Okay, we'll also talk about the nuclear envelope, nucleolus, and the chromatin. We will identify the role of nuclear pores. So, the nuclear membrane consists of some pores. Nuclear envelope or nuclear membrane, that's the same thing. These consist of some pores. These pores are involved in the molecular transport. We'll talk about this also. We will talk about the microbodies also. Within these microbodies, we'll be talking about the peroxisomes and glyoxysomes. We'll talk about the structure and the metabolic functions. Okay, we will also understand the significance of these organelles in the cellular processes. So, these organelles play a very crucial role. So, we'll talk about their functions as well. Okay, so in this session, we'll be talking about the nucleus, which is the NCERT section 8.5.10. We'll be talking about the microbodies. Then we will summarize this entire session. We'll discuss some NCERT based questions related to these topics. There will be some keynotes that will have some interesting questions also. Okay, and some interesting information as well. And then there will be some interesting questions in the end that will enhance your critical thinking ability. Okay, so let's start with today's session. We'll be talking about the nucleus first. So, nucleus is the cell's control center. Nucleus is controlling all the cell, like your nervous system, your central nervous system, which includes brain and spinal cord, that is controlling the whole body. So, nucleus is controlling all the cell activities. So, that's why it is known as the cell's control center. Now, the nucleus was first described by Robert Brown as early as 1831. Okay, so first the cell was discovered by Robert Hooke, but he studied the dead cells. Then Leeuwenhoek, he studied the living cells. Okay, so he observed the RBCs also. Okay, the cells of the spirogyra. Okay, sperm cells as well. Then the nucleus was discovered by Robert Brown. Now, the material of the nucleus, when it is stained by a basic dye, okay, it was named as chromatin by Fleming. So, nucleus consists of the genetic material, which is the DNA. Okay, so this, you can say, DNA is present in very condensed form. If we talk about the eukaryotic DNA, what is the length of the eukaryotic DNA? Okay, so length of eukaryotic DNA is around 2.2 meters. And this DNA is confined in a very little space, which is the nucleus, which you cannot see also. You cannot see cell. So within this cell you have a nucleus, and within this nucleus you have the DNA, which is 2.2 meter in length. So how this DNA is present in such a small space? So this DNA is present in very condensed form. Okay, this DNA is wrapped around the proteins known as histones. So there is a histone octamer around this DNA is wrapped. So negatively charged DNA is wrapped around the positively charged histone proteins. And this unit is known as a nucleosome. Okay. So this nucleosome consists of around 200 base pairs. So many nucleosomes, they are connected together to form the chromatin. So you can see they are talking about the chromatin. So there was some material which was stained by the basic dyes. Okay, like crystal violet. So this basic dye was able to stain this material, and this material was named as chromatin. So what is chromatin? This is the condensed form. So nucleosome, they combine together to form this chromatin. And this chromatin, they make up the chromosomes. Okay. Here you can see, this is the image showing you structure of the nucleus. You can see this is the entire nucleus. Now the fluid present within the nucleus, this is known as the nucleoplasm. Okay. Then within the nucleus, nucleolus is present. We'll talk about the function of the nucleolus also. So this nucleolus basically is involved in ribosomal RNA synthesis, and it is also involved in ribosome assembly as well. Okay. Then you can see this is the nuclear membrane, or you can call it as nuclear envelope. So the eukaryotic organisms, they have a true nucleus. Means the nucleus is surrounded by the nuclear envelope. Okay, this nuclear membrane consists of some pores known as the nuclear pores, through which the molecules can enter and it can leave the nucleus. Okay, so after transcription also, what happens? From DNA, RNA is synthesized. RNA can also travel into the cytoplasm through these pores, and then it is translated. Okay, so some proteins also, which are made by the nucleus, which are coded by the nuclear genome, they can also leave nucleus and they enter into the cytoplasm. Okay, now let's talk about the components of the interphase nucleus. So the interphase is a stage. You must have studied about the interphase stage during the cell division, mitosis and meiosis. Okay, so there is interphase, prophase, metaphase, anaphase. We'll be talking about the interphase nucleus. Okay, so it consists of the nuclear envelope. So nuclear envelope is double membraned outer boundary of the nucleus. Okay, like mitochondria and chloroplast, they have double membranes. They have two membranes, outer and inner. So nuclear membrane is also double membraned outer boundary of the nucleus. Then the nucleoplasm is present within the nucleus. So nucleoplasm is basically the matrix inside the nucleus, the fluid inside the nucleus. Then the chromatin is also present within the nucleus. Chromatin is a network of nucleoprotein fibers. Means chromatin consists of the nucleic acid as well as the proteins. Proteins are also associated. Which proteins? Like histone proteins are associated. So I've told you that the chromatin is made up of nucleosome. Nucleosome is what? Nucleosome is a DNA wrapped around the histone proteins. Okay. So you can call nucleosome as a nucleoprotein. These nucleosomes, they are attached together to form this chromatin. So chromatin will be a network of nucleoprotein fibers. Then nucleolus. Nucleolus is a spherical body within the nucleoplasm. Okay. Nucleolus is involved in ribosomal RNA synthesis. Now there are some exceptions also. Some cells, they lack nucleus. Okay. So nucleus is absent in some cells when they are mature. Okay, that's why they are written nucleus is absent in some mature cells. What are the examples? Example is mammalian erythrocytes. Erythrocytes are also known as RBCs, red blood cells. So initially, these RBCs, they have the nucleus, but when they mature, these RBCs, they lack the nucleus. Okay, why do they lack nucleus? So that there is more space for the oxygen transfer. So more space is occupied by the hemoglobin, to which the oxygen can attach. Then there are some cells present in the plants, which are known as the sieve tube cells. So these sieve tube cells also lack nucleus. Okay, so these are associated with companion cells which help these sieve tube cells. Okay, for more information you can read about the phloem. Okay, clear? Which is a part of the vasculature of the plants. So these two cells lack nucleus. Again, here you can see this is the structure of the nucleus. Okay, here you can see this is the nucleoplasm, which is the matrix. Then nucleolus is a present. This is a region within the nucleus. Here you can see these are the nuclear pores in the nuclear membrane. This is the nuclear envelope, also known as nuclear membrane. Here these are the chromatin fibers. So these are the, what is chromatin? This is the DNA in the condensed form. Okay, let's talk about the nuclear envelope and the nucleolus. Okay, so let's talk about nuclear envelope. So it is a double-layered envelope with a space between called the perinuclear space. So there is a space between the outer nuclear membrane and inner nuclear membrane. This is known as the perinuclear space. The outer membrane is usually continuous with the endoplasmic reticulum. So we have already discussed there is an organelle which is in close association with the nuclear envelope. It is attached to the nuclear envelope. Okay, which is that organelle? That is endoplasmic reticulum, ER. So endoplasmic reticulum is continuous with the outer nuclear membrane. Okay. Now this nuclear envelope is interrupted by minute nuclear pores, which act as a passage for RNA and the protein molecules. So see, DNA is transcribed into RNA. Then this RNA will travel through this nuclear pores and it will enter into the cytoplasm. Okay. Some proteins also, they move from the nucleus into the cytoplasm. Okay, so these pores allow the transfer of the molecules between the nucleus and the cytoplasm. Okay, there are some proteins which are produced in cytoplasm also. These are transported inside the nucleus also, but these are just very few proteins. Okay, now when the RNA it reaches the cytoplasm, then it can be translated into the proteins. Okay, here you can see in this image, this is the endoplasmic reticulum, which is in close association with the outer nuclear envelope. Okay, then you have the inner membrane. Between these two, there is a perinuclear space. Okay, this is the perinuclear space, you can see. These are the nuclear pores. Okay, these are the pores. Clear? This is the nuclear envelope, you can see. Outer nuclear envelope and inner nuclear envelope. Here you can see this is the chromatin. Chromatin is what? It is nucleoprotein, means DNA along with the associated proteins like histones. So DNA is negatively charged, histones are positively charged because they consist of basic amino acid like lysine. Clear? Now let's move on. Let's talk about the nucleolus. What is nucleolus? So it is a spherical, non-membrane bound. So there is no membrane around nucleus and it is found in the nucleoplasm. Now its content is continuous with the rest of the nucleoplasm. So nucleolus is just a region present within the nucleoplasm. It doesn't have any boundaries. So the content of the nucleolus, it is continuous with the rest of the nucleoplasm. So it is very similar to like nucleoid. There is no boundary of the nucleoid region in the prokaryotes. Okay. So nucleolus, what is the function? So it is a site for active ribosomal RNA synthesis. Okay. So synthesis of ribosomal RNA, this is done within the nucleolus. And we all know that ribosomes, they are made up of ribosomal RNA. Ribosomes have two subunits, smaller subunit and larger subunit. Okay. So ribosomal RNA synthesis is being done within the nucleolus. So here you can see, this is an image showing you nucleolus. This is a nucleolus, which is a region present within the nucleus. And it is, you can say, found in the nucleoplasm. Okay, now let's talk about the chromatin and the chromosomes. What is chromatin? So it is a loose and indistinct network of nucleoproteins. Nucleoproteins means nucleus and proteins. Okay, see that these are nucleoprotein fibers seen in the interphase nucleus. Okay, then it contains DNA. Okay, they are specifically writing interphase nucleus here because this chromatin, this changes, the condensation level changes in the other stages: prophase, metaphase, anaphase, telophase. Okay, later on you have chromosomes. Okay, so there is condensation and decondensation in various stages. So they are specifically talking about the interphase nucleus. So chromatin contains DNA, which is a nucleic acid, and basic proteins called histones. Histones are positively charged. Along with the histones, some non-histone proteins are also present and some RNA is also present. So this is what chromatin is made up of. So basically, these are the three components: DNA, histones and non-histone proteins. Okay, so here you can see this is an image showing you the chromosome structure, and the chromatin also they are showing. Chromatin is also shown to you. Okay, this is the chromosome. Chromosomes are made up of chromatin. Okay, clear? Chromatin is also associated with some proteins. Here you can see this is a dividing chromosome. Here you can see this is better image. This is the entire chromosomes. Chromosomes are made up of this chromatin. Now they have enlarged this section here. Okay, chromosome consists of these chromatids. Okay, if you enlarge it, you can see these are the nucleosomes. These nucleosomes, they are attached together to form the chromatin. Here it is written chromatin. Okay, this is the DNA wrapped around the octamer, histone octamer. Histones are basic proteins. They are positively charged. DNA is negatively charged. Okay, then this is another nucleosome, another nucleosome. So when you view it under the electron microscope, it will be seen like beads on a string. Okay, so this is how the packaging of the DNA is done in eukaryotic cell. This packaging of DNA is different in prokaryotic cell. Okay. Okay, let's talk about the chromosomes now. Now, during cell division, chromatin condenses to form structural chromosomes. So, see, first you have nucleosomes. Nucleosome combine to form chromatin, and chromatin condenses further to form chromosomes. Okay, each chromosome consists of a primary construction which is known as the centromere. So, the chromatids, a chromosome have two chromatids. Okay, these are known as daughter chromatids. So these chromatids, they are attached at a region known as the centromere. Now, this centromere consists of disc-shaped structures called the kinetochores. So these kinetochores are present on either side of the centromere. And the spindle fiber, they attach to the kinetochores present within the centromere. Okay, so they don't attach to the centromeres, spindle fibers. They attach to the kinetochores, which are the regions present within the centromeres. And then separation of the chromosome occurs. So here you can see this is the entire chromosome. These are the chromatids. Okay, these are the daughter chromatids or sister chromatids. Okay, sister chromatids, not daughter chromatids. These are the sister chromatids. Okay, this is a centromere, this region. And you can see within the centromere, kinetochores are present. These are the regions present within the centromere, and here the spindle fibers will attach. Now let's talk about the type of chromosomes. Now, based on the position of the centromere, the chromosome can be classified into four types. Now, what are the four types of chromosome based on the position of the centromere? First is metacentric. So, in metacentric, centromere is in the middle, forming two equal arms. Okay, then you have the submetacentric. Here, the centromere is slightly away from the middle, resulting in one shorter and one longer arm. Okay, so here this is the metacentric, this is submetacentric. In metacentric, the centromere is in the middle. Okay, in submetacentric, centromere is slightly away from the middle. So that's why one arm is shorter, one is longer. So this is the shorter arm up on the you can say above side, and this is the longer arm on the lower side. Short arm is known as the P arm and long arm is known as the Q arm. Remember this. Okay. Now next type of chromosome is acrocentric. So in acrocentric, what happens? The centromere is situated close to its end, forming one extremely short and one very long arm. Okay, then there is telocentric. Here the centromere is terminal. Okay, so this is acrocentric chromosome. Here the centromere is situated close to the end, but it is not at the terminal position. When the centromere is at the terminal position, it is known as the telocentric. Okay, now sometimes in chromosomes, secondary constrictions are also present, which you can say are known as the satellites. Okay, so let's talk about the satellite. DNA. So some chromosomes have a non-staining secondary constriction. So primary constriction was known as centromere. Some chromosomes also have the secondary constriction. Now this secondary constriction gives the appearance of a small fragment called the satellite. Okay, so this is a secondary constriction. These are known as satellite DNA. Okay, so this was about the nucleus and the chromatin and the chromosomes. Now let's talk about the microbodies. So, membrane-bound minute vesicles found in the cytoplasm, this is known as the microbodies. Okay, they contain various enzymes for specific metabolic activities. Metabolic activities are all your biochemical reactions. They are present in both plants and animals. Here you can see this is an image showing you microbodies. Okay, this is the plasma membrane. Then you have the lipid layer. And this is the crystalline core of the microbodies. These microbodies consist of various enzymes which are involved in biochemical reactions. What are the function of microbodies? First is oxidation. So microbodies are involved in various oxidation reactions. What all oxidation reactions? First is it breaks down fatty acid and removes toxins like hydrogen peroxide. Okay, so hydrogen peroxide is a toxin. Okay, so this is removed. Now it is also involved in the metabolism. So they play a role in diverse metabolic pathways. Now plant use photorespiration and convert lipids to carbs during the seed growth. So this is occurring in these microbodies. Okay, during seed growth, carbohydrates are required. So lipids are converted into carbohydrates during the seed growth. This will be done in the microbodies. Okay, initially, seeds, they are born inside the seed, inside the soil. Okay, so they cannot photosynthesize. So they need to survive for some time. They need to germinate. Then they form the plantlet and then they photosynthesize. Okay, so initially carbohydrates are required for the seed germination. So lipids are also converted into carbohydrates. Next is detoxification. So peroxisomes, which are a kind of microbodies, these peroxisomes help detoxify harmful substances as well. Okay. Here, this is the image showing you microbodies. So you must have studied about the process of photorespiration. This involves peroxisomes. Okay, it involves three organelles: mitochondria, chloroplast, and peroxisomes. So you should read that and then you will be able to understand this process of photorespiration. Examples of microbodies. First is peroxisomes. So these peroxisomes are involved in metabolic processes including breaking down fatty acid and detoxifying the harmful substances. These peroxisomes contain enzymes that produce and degrade hydrogen peroxide as well. Next one is glyoxysomes. It is also a kind of microbody. So these are specialized peroxisomes found in the plants, particularly in the fat storage tissue of the germinating seeds. So where are glyoxysomes present? They are present in the fat storage tissues of germinating seeds. Here you can see in this image on the left hand side, this is of peroxisome. Okay, here you can see they consist of these enzymes. Clear? They have a crystalline core. They have a lipid bilayer, you can see. Then these are the glyoxysomes. Okay, they also consist of this crystalline core. Okay. Now enzymes present in the peroxisomes, they help in the breakdown of the fatty acid also. Clear? Peroxisome is also involved in photorespiration. Let's talk about peroxisome first. Peroxisomes are membrane-bound organelles found in the eukaryotic cells. Okay, they are single membrane. There is only one membrane around the peroxisomes. Okay, like mitochondria have two membranes, here they have only single membrane. So peroxisomes are surrounded by a single lipid bilayer. Like plasma membrane has a phospholipid bilayer, these peroxisomes also, they have a single lipid bilayer. They have a granular matrix. So inside the membrane is a matrix containing various enzymes, and they have a core also in the center, crystalline core. Now, about the size and shape of the peroxisomes. So mitochondria are usually round structures that vary in size based on cell needs. Clear? So in photorespiration, mitochondria, chloroplasts and peroxisomes are also involved. Okay. So here you can see this is an image of the peroxisome. They have a crystalline core also. They have enzymes also. These enzymes are involved in metabolic functions. Okay, function of peroxisomes, fatty acid oxidation. This we have seen. So peroxisomes play a crucial role in the breakdown of long chain fatty acids through a process called beta oxidation. Hydrogen peroxide detoxification. So they contain the enzyme catalase. So catalase is very important enzyme which converts the toxic byproducts hydrogen peroxide into water and oxygen. So this hydrogen peroxide, they all act as radicals and they can destroy the tissues. They can cause damage to the tissues. So this hydrogen peroxide needs to be removed. So catalase can convert this hydrogen peroxide into water and oxygen. Okay. We're talking about the function of peroxisome. Next is photorespiration. In plants, peroxisomes are involved in the process of photorespiration, which helps to metabolize byproducts of the photosynthesis. Okay, you will read about photorespiration in the chapter photosynthesis. Okay, there you will learn about C3 cycle, C4 cycle. Okay, you will also learn about the photorespiration as well. Clear? So here there three organelles are involved. It is also involved in some other metabolic processes. They also participate in the synthesis of bile acid, cholesterol, and plasmalogens, which is a type of phospholipids. Okay, these are some additional functions. Okay, you should remember the main functions. Glyoxysomes are similar in structure to peroxisomes, being spherical or oval shaped. The structure range from 0.2 to 1.5 micrometer in diameter, and they are bound by a single lipid membrane. Okay, so microbodies, peroxisomes and glyoxysomes, they are bound by just a single lipid bilayer membrane. Now the matrix, which is the interior of the glyoxysomes, contain enzymes for both the glyoxylate cycle and other peroxisomal enzymes. So within the glyoxysomes, glyoxylate cycle occurs. Okay. Now these are synthesized by endoplasmic reticulum. Then what are the function of glycosomes? The primary function of glyoxysomes is to facilitate the conversion of stored fat into carbohydrates through the glyoxylate cycle. Okay, so fats is being converted into carbohydrates. Then carbohydrate can be used as a respiratory substrate. This cycle allows the cell to bypass the two decarboxylation steps. First is the citric acid cycle known as the Krebs cycle in the mitochondria. Okay, and preventing the loss of acetyl coenzyme A as carbon dioxide. Okay, so when you will read these cycles, then you will understand this more. Okay, clear? Now next, we're talking about the function of glycosomes. So here instead acetyl coenzyme A, which is derived from fatty acid breakdown, is converted into succinate, which can then be used for gluconeogenesis to produce glucose. Okay, so in glycosomes, glycosylate cycle is being carried out in which fatty acid is being converted into carbohydrates. Clear? Okay, this step is crucial energy source for the seedling. So when there is a seed germination, you require carbohydrates. So carbohydrates are being provided to this seed for germination by this process. Now, glycosomes are most active during the germination of the seeds. See, okay, they are very important for the germination of the seeds and they are quite active also at that time because they are converting fats into carbohydrates. Now, seeds, those rich in fats and oils, for example, corn, soybean, and sunflower, here the glycosomes are, you can say, more in number. Okay, so this was about the nucleus and the microbodies. Clear? Now let's summarize this entire lecture. We talked about the chromosomes and the chromatid. Okay, chromatin. So chromatin is what? This is a condensed form of DNA. It is made up of nucleosomes. So nucleosomes, they are attached together to form the chromatin, and chromatin, it condenses further to form the chromosome. Okay, so nucleosomes, then chromatin, then chromosomes. So chromosomes can be said to be thread-like structures which contain DNA and proteins. Okay, chromatin also consists of DNA and proteins. Chromatin carry genetic information in the cells because they are made up of DNA. Okay. Now there are three main type of there are four main type of chromosomes actually: metacentric, submetacentric, acrocentric, and telocentric. Okay, microbodies are small spherical organelles bounded by single membrane. Okay, they are bounded by single membrane only. They contain various enzymes for specific metabolic functions. So there are different functions of peroxisomes and glyoxysomes. Peroxisomes, they are involved in photorespiration. Glyoxysomes, they carry out glyoxylate cycle. Peroxisomes are specialized microbodies that contain enzymes for breaking down fatty acids and toxic substances like hydrogen peroxide. Okay, then you have the glyoxysomes as well. Peroxisomes also play a crucial role in cellular detoxification, lipid metabolism, and are especially abundant in the liver cells. Glyoxysomes are specialized peroxisomes found primarily in the plant seed, containing enzymes necessary for converting storage fats into sugars during the seed germination. So glycosomes, see, they have a very important role during the seed germination. Both peroxisomes and glycosomes are essential for cellular metabolism and can self-replicate by division of the pre-existing organelle. So they can self-replicate also. Now let's discuss some NCERT based questions. So first question is, the nuclear membrane is a, what are the options? It is a single membrane, it is the double membrane with nuclear pores, it is triple membrane, or fourth, it is non-porous membrane. What will be the correct answer? So let's have a look at the answer. So answer will be, okay, option number B. Option number B is the correct answer, double membrane with pores. Let's have a look at the explanation. So the nuclear membrane, also known as the nuclear envelope, consists of two lipid bilayers, outer and an inner membrane. Okay, it also consists of the nuclear pores that allow selective transport of molecules between nucleus and the cytoplasm. Now the space between the two membranes, outer and the inner membrane, this is known as perinuclear space. Now nuclear pores are basically complex protein structures that regulate the molecular traffic. Now this structure is essential for protecting genetic material also and regulating nucleus cytoplasmic transport. Here you can see in this image, this is the nucleus. The outer membrane is shown to you, inner membrane is shown to you. Now between there is a perinuclear space between outer and the inner membrane. Okay. These are the nuclear pore complex you can see. This nuclear pore complex help to regulate the molecular traffic. Next question, which of the following chromosome is characterized by having the centromere located near the end of the chromosome, resulting in very short arm and very long arm? Okay, so here centromere is located near the end. So there is a difference. They are saying centromere is located near the end, not at the terminal. So what would be the correct answer? Metacentric, submetacentric, acrocentric, or telocentric? Alright, so let's have a look at the answer. So answer is option number C, acrocentric chromosomes. Okay, so what are acrocentric chromosomes? So here the centromeres are located near one end. So this results in very short arm known as the P arm and the long arm known as the Q arm. Now the short arm often contains nuclear organizing region. Example in humans, chromosome 13, 14, 15, 21 and 22. These are examples of acrocentric chromosomes. Okay, this is an image showing you acrocentric chromosome. Here the centromere is towards the end of the chromosome. Okay, and then there is a short P arm and there is a long Q arm as well. Next, microbodies are characterized by options are presence of ribosomes, second is oxidative enzymes, third is storage function, and fourth is transport function. What would be the correct answer? So let's have a look at the answer. Okay, so answer will be option B, and only B is the right answer. Okay, no other option is the correct answer. So they have oxidative enzymes. Okay, examples of microbodies, they are peroxisomes and glyoxysomes. They are single membrane bound organelles. Okay, they doesn't consist of any ribosomes. So ribosomes are not present here. Ribosomes are associated with endoplasmic reticulum. What are their primary function? Primary function is metabolic. Okay, so they have enzymes which are involved in metabolic processes, and their function is not storage or transport. They are basically involved in breakdown of fatty acids and detoxification of molecules such as hydrogen peroxide. Okay, they have catalase enzyme as well for hydrogen peroxide breakdown. They are self-replicating organelles also. Okay, so there is specific function of peroxisomes and glyoxisomes. Okay, next is which of the following statement correctly describe the relationship between glyoxisomes and peroxisomes? So let's read this statement. First statement is glyoxisomes are found in animal cells while peroxisomes are found in plant cell, only in plant cell. Next is glyoxisomes are specialized peroxisomes found in germinating oily seeds containing enzyme for glyoxylate cycle. Option number C is peroxisomes and glyoxisomes are identical structures with the same enzymatic content. And fourth is glyoxisomes are involved in photorespiration while peroxisomes are involved in fatty acid breakdown. What would be the correct answer? Okay, so let's have a look at the answer. Answer is option B. So glycosomes are specialized peroxisomes which are found in germinating oily seeds. Okay, they consist of enzymes for the glyoxylate cycle. Okay, so glycosomes, they are specialized peroxisomes that contain enzyme for glyoxylate cycle, convert stored fatty acid into carbohydrates during seed germination, are found specifically in the germinating oil seeds such as castor seeds. Now, they share some enzymes with the peroxisomes as well, but have additional specialized enzymes as well. Okay, now let's discuss some keynotes. We talked about the nucleus. It is the largest organelle in the cell. Okay, we're talking about the animal cell. Otherwise, chloroplast is the largest. It contains DNA in the genetic material, then has double membrane around it, controls the cell activity. Okay, it is self-replicating also, contains nucleolus. Let's talk about microbodies. They are small membrane-bound organelles, include peroxisomes and glyoxisomes, contain oxidative enzymes, help in detoxification, important for lipid metabolism, and they are single membrane-bound structures. So they have single lipid bilayer. Now there are some interesting questions for you. First is how do proteins, you can say, which are larger than the nuclear pore size enter into the nucleus? Next one is why do some cells have multiple nuclei while others have none? So some of the cells they doesn't consist of any nucleus, whereas some cells they are senocytic, they consist of multiple nucleus. Next is what happens to the nuclear envelope during the cell division, and what is the relationship between peroxisomes and photorespiration in the plants? Okay, so by photorespiration in plants, what happens? The carbon dioxide which is fixed, that is lost in the form of carbon dioxide. Okay. Now next is why are peroxisomal disorders often fatal? Okay, so these are some questions. Okay, which you have to think about. You have to critically analyze these questions. Okay, so we have discussed the theory now. Now let's discuss the DPP related to these topics. Okay, what is the first question here? Which of the following statements about the nuclear envelope is incorrect? Means which is false. So option A, it consists of two parallel membranes with a perinuclear space in between. Next is the outer membrane is continuous with the endoplasmic reticulum and BS ribosome. Third is nuclear pores are formed by the fusion of two membrane allowing bidirectional transport. And fourth is the nuclear envelope remains intact and uninterrupted during the cell division. What is the correct answer? Okay, here the correct answer will be option number D. Nuclear envelope remains intact and uninterrupted during the cell division. This is the false statement. Rest of the statements are correct. Next one, what is the primary function of nucleolus? DNA replication, synthesis of ribosomal RNA, storage of histones, and formation of the nuclear pores. Alright, so here the correct answer will be option number B. Okay, synthesis of ribosomal RNA. This is done within the nucleolus. So option number B is the correct answer. Now let's move on to the next question. Which of the following cells is anucleate? Means it lacks the nucleus. Hepatocytes mean liver cells. Mature erythrocytes in humans means RBCs. Okay, they're talking about mature RBCs. Then neuron, which is nerve cell, and then sperm cell. What is the correct answer? Okay, RBCs is the correct answer. Means option number B. Erythrocytes are also known as RBCs. So mature RBCs, red blood cells, they lack nucleus. So they can efficiently transport the oxygen. Okay, so option number B will be the correct answer. Next one, chromatin is composed of. Options are DNA and histones only, DNA, histones, and non-histone proteins. Third is DNA and RNA only, and fourth is RNA and histones only. What will be the correct answer? Okay, option number B will be the correct answer here. Chromatin is composed of DNA, histones which are positively charged, and non-histone proteins. Next one, which of the following type of chromosome is the centromere located terminally? Okay, what is that type of chromosome called in which the centromere is terminal? Options are metacentric, submetacentric, acrocentric, and telocentric. What will be the correct answer? Okay, telocentric is the correct answer. Means option number D. Clear? Here the centromere is terminal. Alright, so we have discussed the DPP also. Okay, all our answers are correct. So with this, we end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.